corner here.
Well, it's an iPad.
And we're going to call it prophase. Okay? So in the first quadrant up at the top,
black.
you want prophase. Thank you, Ryan.
Yeah.
Okay? First phase, right? PMAS. Everything that I do up here should end up on your papers. Okay? So now I want you guys to draw a nice big circle. Take up the majority of the square. Okay? Now, cells are not perfect, so your little circle does not need to be perfect. Okay? So we've drawn a nice circle.
Blood cell?
Huh?
Are we drawing red blood?
If you want to pretend that this is a blood cell, you can. This is just a generic cell.
So it's just the
But it's important what type of cells go through mitosis. What vocab term did we learn yesterday and the day before? Body cells. What's a fancier name for
somatic?
Somatic. Awesome. Okay, so maybe in the corner I'm going to remind myself that this is with somatic cells.
I just want it back.
Okay, which as Audrey and Lex were saying is also known as a body cell. Okay, so a really good example could be like your skin cell.
Can I put some parentheses be body cell?
Sure, if you like to. Okay, all right. Next thing that we need to draw, we are going to draw a huge nucleus.
Oh, sweet.
Okay, just to give ourselves some space. We know that a nucleus doesn't take up the whole inside of the cell.
It's a little circle.
Right. But for diagram reasons right now, we're going to make it a little bigger. So I want you guys to draw a dotted line a little bit inside the solid line we just drew. Okay? Philip.
Are we trying to circle with our pencil or a color?
Everything in black is in pencil. Very fun.
Okay. Ignore pencil. Right? So
Is this big enough?
I would draw a little bit bigger. Okay? The reason that we have drawn it as a dotted line is on purpose. Okay? We are trying to represent, and you can draw this wherever you guys have space, okay? That the nucleus is disappearing. If I can spell disappearing right.
Is it big enough?
Okay, good. So that nuclear membrane that exists in G1, S, and G2 is starting to disappear. Okay? During mitosis, it does not exist. Right? We also are going to draw.
But does it reform after a while?
Great question. Yes, it does.
Okay.
Yes, it does. Right before we split, it starts to reform. Okay? I'm going to draw two circles on opposite sides of my page. Okay? Inside my cell. And I'm going to draw little squares in them. And I'm going to draw some of these fibers coming out of them. Does anybody want to take a stab at what any of these things are called?
Antenna.
Okay, so one at a time. Philip has his hand raised.
Are the fibers called spindle fibers?
That they are. My spindle fibers start to form. I'm just going to call them spindles here. Okay. Philip? Harvey?
Yeah. Is this when they pull away, creating another one?
They are going to start to do that. This is starting that whole process, absolutely. Okay. Does anybody remember from our vocab words the other day? Okay. Okay, so we'll have to make sure we catch up with that. What the microtubules that the spindles grow out of are called? Starts with a C. Centrioles. Awesome, Lex. Okay, so these little squares are representing centrioles. Okay, we're not going to label these in every picture, just this first one. Okay, those are all centrioles. And last but not least, what is the organelle that holds the centrioles? Sounds just like another word that we already knew.
Centrosome.
Centrosome, awesome. So there's a lot of labeling in prophase, but once we have labeled it once, we're not going to label it in every single phase. Okay? Awesome. Now, wait, we're going to draw one more thing and then we're going to use our colors. Okay? We talked about the word diploid yesterday.
Diploid.
Does anybody remember what diploid means? Shanice?
I think it was two haploids.
Okay, 2 haploid sets. We mentioned how that essentially means we have a full set of DNA. Awesome. Does anybody remember what the number and the letter that represent diploid mean?
The letter is N.
Yep.
2.
Very close. 2N. 2N. Okay.
The same thing.
We always write the number first.
Oh, okay.
Okay, 2N. It is kind of like math. Okay, N is representing a variable, the amount of chromosomes. So in our picture, we're going to draw a little cell that has 3. So our diploid number in this cell is 3. Okay, now choose 3
Yeah.
colors.
I feel like I'm in math now.
You're in math now.
It's
Miss
2N.
Please don't be mad at me, but
Variables.
this pen rolled off my desk and it
Oh,
broke.
it broke.
Oh,
Audrey, how
don't
could
be
you?
mad.
It's okay.
I feel so
All right,
bad.
so in the center part, you're going to use each of the colors to draw a chromosome. Okay, we're going to draw in the sister chromatid form.
I'm very bad at
Suck,
these, so
but
I
I
know.
know.
You can just draw X's if you want. X's are totally acceptable. Okay, so they're just starting to condense. They're just kind of hanging out as the nuclear membrane breaks down. Awesome. Okay, so I have 1, 2, 3 chromosomes. Okay, we remind ourselves that each one of these, as Ryan mentioned earlier, is actually representing sister chromatids. It's a nice chromosome, maybe. Okay. So if your chromosome skills aren't up to par, you can just draw them as X's for
I
now.
wrote Chrome with
Can you
this.
stop again?
Oh, that's okay. 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 So finish coloring those in. How are we doing? We're almost there. For those of you that are finished, we're going to start drawing the next one, okay? We want to draw our